Let's talk about functional groups. More importantly, let's talk about why they matter. Functional groups are small, recognizable clusters of atoms that give molecules predictable properties. Spotting these groups helps us understand everything from enzyme activity to cell structure. Most biology classes will cover 7 functional groups: hydroxyl, phosphate, sulfhydryl, carbonyl, amino, carboxyl, and methyl groups. Because we want to focus on biologically important functional groups, we're going to add ester for lipids and acetyl for DNA, bringing us to 9. Besides the specific functions that I'll describe, many of these serve to assist in molecular interactions through hydrogen bonding or hydrophilic interactions. For example, how do enzymes and substrates interact? What about enzyme inhibitor interactions? How do proteins hold their tertiary structure? In many cases, these interactions are due to the functional groups we'll discuss. The hydroxyl group is composed of a single oxygen and a hydrogen. The oxygen makes the molecule polar, so these are soluble in water. The most important examples in biology include alcohols like ethanol and sugars. Phosphate groups consist of a central phosphorus atom and 4 oxygens. Since the oxygens can carry multiple negative charges, it takes a lot of energy to hold multiple phosphates together. Breaking this bond is how ATP transfers energy, and forming this bond is the main purpose of cellular respiration. Adding a phosphate group to a molecule known as phosphorylation tags it and turns proteins on or off. Sulfhydryl groups pair to make disulfide bridges. These are very strong covalent links that stabilize folded proteins. Carbonyl groups are composed of a single double-bonded oxygen. When at a chain end, it's called an aldehyde. Mid-chain, it's called a ketone. Carbonyl groups are polar and important for chemical reactions that add or eliminate things from molecules. The amino group is basic, meaning it grabs hydrogen ions from solutions. Amino groups make up one side of the core structure of amino acids. The carboxyl group looks like kind of like a combination of a ketone and a hydroxyl. It's acidic, meaning it donates hydrogen ions to solutions. Carboxyl groups make up the other side of the core structure of amino acids. They're also important in fatty acids, where they participate in dehydration synthesis reactions. Esters form when carboxyls react with hydroxyls. For example, a fatty acid and a glycerol combine through dehydration synthesis, forming an ester. This is seen in both triglycerides and phospholipids. Methyl groups are composed of a terminal carbon and 3 hydrogens. This is the only group we're covering that's hydrophobic. Adding a methyl group to DNA, known as methylation, tightens histone proteins and silences genes. Smoking-induced hypermethylation of tumor suppressor genes is one factor promoting lung cancer. Acetyl groups are like a combination of a ketone and a methyl group. Adding acetyl groups to DNA, known as acetylation, loosens chromatin to activate genes. Acetyl also shows up in the structures of NAG and NAM, the components of peptidoglycan in bacterial cell walls. Let's build some NAG from a simple glucose molecule. Here's the structure of glucose. Notice the hydroxyl groups attached to the carbons. But there aren't any amino groups or acetyl groups, just hydroxyl for now. By adding an amino group, this molecule's name changes to glucosamine. Finally, we can add an acetyl group to that amino. Doing so changes the molecule's name to N-acetyl glucosamine, or NAG. The N-acetyl part of N-acetylmuramic acid is the same, an acetyl group attached via an amino group. NAG and NAM are core components of peptidoglycan in the cell walls of most prokaryotes. Hydroxyl, phosphate, sulfhydryl, carbonyl, amino, carboxyl, ester, methyl, acetyl. Spot these in any molecule to predict its behavior.